Hi, in this video, we'll be learning about the stages of the lytic cycle. Let's get started. There are 5 stages in the lytic cycle. The 5 stages are attachment, penetration, biosynthesis, maturation, and release. The first step is attachment. In the attachment phase, the bacteriophage, or the virus that can only infect bacteria, attaches to the cell wall of the bacteria. This happens because the bacteriophage can bind to the unique receptors found on the bacterium that it is able to infect. So, whenever you hear your professor talk about the specificity of a virus, this just means that a virus can bind to specific receptors on a specific type of cell. And this means that not every virus can infect every bacterial cell. The next stage is penetration. In this stage, the bacteriophage is able to penetrate through the cell wall of the bacterium, in this case, the host cell, and injects its DNA into the cytoplasm of the bacterial cell. And it is able to penetrate the cell wall of the bacterial cell by using its lysosome to degrade into the cell wall, thus creating the hole, which would allow it to inject its genome or its DNA or RNA into the bacterial cell. The next stage is biosynthesis. Once inside the cytoplasm, the viral DNA disrupts. This disrupts the host's normal activities. And what I mean by that is instead of the host or the bacterial cell making its own protein, its own RNA, its own DNA, what the bacterial cell is going to do now is produce viral particles. It's going to be replicating the bacteriophage's DNA. It's going to be making the head of the bacteriophage, the tails, is going to be making the components needed to build a bacteriophage. The next stage is maturation. In the maturation stage, what happens is that the components of the bacteriophage that were created in the previous step or in the previous stage, which was biosynthesis, these parts now are put together to form full or complete viruses, in this case, the bacteriophage. So the fully assembled bacteriophage is also called a virion. The last step is the release step, and these virions use their lysosome to break down the cell wall in order to escape or to release. And what ends up happening is it causes the bacterial cell to die or to be killed of osmotic lysis, because by breaking through their cell wall, that reduces the strength of the bacterial cell's cell wall, which makes it more susceptible to osmotic lysis, aka water rushing into the bacterial cell and the bacterial cell wall not being strong enough to resist that. And so you can think of it as water rushing in and causing the bacterial cell to blow up or explode. And once they're outside of the bacteria, they can go on and infect new bacterial cell, essentially restarting this whole process again. So really quickly, let's review what we've learned in this video. In this video, we've learned about the 5 stages of the lytic cycle, and the 5 stages are attachment, penetration, biosynthesis, maturation, and release. In the attachment stage, the bacteriophage, or the virus that can only infect bacteria, it attaches to the cell wall of the bacteria. Next is the penetration stage. In this stage, the bacteriophage, having been attached to the cell wall of the bacteria, creates a small hole in order for it to insert its genome into the bacterial cell. Next up is the biosynthesis stage. In the biosynthesis stage, the bacterial cell can now produce the viral particles needed to create new viruses. The next stage is maturation. In this step, the viral pieces that were made in the biosynthesis stage are put together. Last is the release step. In this stage, the new bacteriophages created, or the virions, use their enzymes to break through the cell wall of the bacteria in order to exit the bacteria and go on to infect new bacterial cells. I hope this video helps. Please like, subscribe, comment, and share this video.